Warm good afternoon to one and all. It's my pleasure to have the Best Researcher Award in International Conference on Genetics and Genomics of Disease. Myself, Dr. Ritesh Bhole, I'm working as a professor at Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research, Pimpri, Pune. Today, I'm discussing my topic named as Unlocking the Potential of PROTAC, a comprehensive review of protein degradation strategies in disease therapy. As we all know, the PROTAC is not the new concept. The proteolysis targeting chimeras are the groundbreaking class of molecules in the field of drug discovery and development. They are designed to selectively degrade specific proteins within the human body. PROTAC work by hijacking the cellular machinery which is responsible for protein degradation, such as ubiquitin protease system. The key innovation behind the PROTAC is their ability to target and eliminate disease-causing protein, which traditionally small molecule often cannot do effectively. This targeted protein degradation approach holds a great promise for treating various diseases, including cancer and certain genetic disorders. Traditionally, drug development primarily focusing on inhibiting the activity of disease-causing protein. However, some proteins are challenging to inhibit directly due to their structure and functions. To overcome this, we come up with a newer technology named as proteolysis targeting chimeras. So this PROTAC emerged as a solution to this challenge by taking an alternative route. They aim to degrade the problematic proteins. PROTAC leverages the ubiquitin protease system, a natural cellular pathway which is responsible for degrading unwanted or damaged proteins. This system involves tagging protein with ubiquitin molecule, marking them for degradation by proteasome, a cellular garbage disposable system. So when we see the structure of this PROTAC, it is a hetero bifunctional small molecule. It consists of two parts, which is covalently linked to each other. One part is capable to engage E3 ubiquitin ligase, known as anchor, while as the other part binds to the protein of interest known as warhead. It helps in degradation of untriggable protein through ubiquitinase reactions. So this is the background about the PROTAC technology. Now we are going to see the major milestone for development of the PROTAC. The development of proteolysis targeting chimeras has marked by the several major milestones, reflecting the increasing recognition of their potential in the drug discovery and progression of this field. Here are some of the key milestones of the development of the PROTAC technology. This concept is coined in 2001. After the research work which is proposed by Craig M. Graves and the co-workers, introducing the idea of using small molecule to induce the degradation of specific target molecules. In 2008, further advancement has been takes place, but it is commonly it is known as the proof of concept. In that particular proof of concept, a significant milestone was successfully demonstration of PROTAC technology effectiveness in degradation of androgen receptor known as AR, a protein which is associated with the prostate cancer. This proof of concept study provide a critical evidence of the technology and its feasibility. Further advancement has been done in 2010, where there is a requirement of the PROTAC design structure has been takes place. Throughout the 2010, researcher made a significant strides in redefining the PROTAC design principles, including linker optimization and development of various ligands for target proteins and E3 ubiquitinase ligase. This period marked as a maturation of the PROTAC technology. The advancement further takes place in 2015, whereas PROTAC with the VHL and CRBN small molecular ligands are being proposed. Actually, in 2019, the first clinical trial on PROTAC has been proposed and undertaken by the Arvinas, which is a pharmaceutical company which initiated the first clinical trial of the PROTAC-based technology. Their lead compound ARV-110 entered into the clinical trials for treatment of patients with metastatic castrate-resistant prostate cancer. So in future, that molecules may come into the market. Further advancement has been takes place in 2020 when the expansion of this PROTAC concept into the drug discovery has been takes place. In 2021, advancement in the targeted protein degradation, which is further advancement by finding of new E3 ligase molecules. Continuous innovation has been takes place in this field, and might be in an upcoming future. PROTAC may be the solution for many life-threatening diseases such as the cancer or any viral diseases and some infectious diseases also. Next, we are talking about how this PROTAC is being acted. What is the exact mechanism of action of this PROTAC? So, as we seen here, the PROTAC molecule is going to bind with a target. So that binding with a target is known as protein of interest. As we already said that the PROTAC is very much specific for the protein, so it goes and going and bound to the protein of interest only. The process begins with the binding of PROTAC molecule to the protein of interest. Then there is a formation of ternary complex. Once bound with the POI, the PROTAC molecule form a complex by binding to the E3 ligase. This complex effectively brings the POI and E3 ligase into the close proximity. After that, the ubiquitination reactions of this POI has been takes place, which is the ternary complex E3 ligase tag the POI with the ubiquitin molecules. This tagging or ubiquitination marks the POI for the degradation. So other proteins remain as a separate one, and the proteins which is going to degrade is only protein of interest. So finally, the protein which is tagged by our PROTAC molecule is recognized by the proteasome. A ubiquitinated POI is then recognized by the proteasomal degradation system, and finally the degradation occurs. This highly specific and targeted mechanism of action enables PROTAC to selectively degrade specific protein of interest, while sparing other cellular proteins remain untouched. It represents an innovative approach to address challenging disease-causing protein and holds potential for development of more precise and effective therapeutic interventions. So as we've seen in this mechanism of action, here is the more simplified mechanism of action. So simplified schematic representation of ubiquitinase mechanism. So as we already discussed, that protein target, which is POI, start with the protein of interest that needs to be regulated or degraded. Ubiquitin molecules are small molecules that plays a crucial role in the ubiquitination process. So these are the two different capable fundamentals of this PROTAC technology. Then even enzyme activation is being occurred. The even enzyme activation is the process which begins with the activation of ubiquitin by an even enzyme. This activation involves attaching of ubiquitin to the even enzyme. After activation of even enzyme, activated ubiquitin is then transferred to the E2 enzyme, which is known as ubiquitin conjugating enzyme. After that, it reaches to the E3, and E3 ligase enzyme recognize both POI and ubiquitinase charge E2 enzyme. After that, finally, ubiquitination reaction takes place. This ubiquitination is nothing but the marking of the PROTAC with the protein of interest. So this process may be repetitive again and again, and there may be formation of many polyubiquitin molecules. There's many formation of many polyubiquitin molecules are known as polyubiquitinin chain. This chain is finally recognized by the proteasome. So proteasome degradation system is going to recognize that polyubiquitin chain, and finally it leads to the degradation. The proteasome or other degradation machinery processes polyubiquitin POI breaking into the smaller fragments. This ubiquitination process plays a critical role in regulating the levels and functions of protein within the cells, and it can be ultimately lead to the degradation of specific protein when they tag with the ubiquitin. So this is the whole systematic representation of ubiquitin mechanism. Next, we are discussing about the difference between the PROTAC and SMI, that is small molecule inhibitors. So as we already discussed that PROTAC is going to affect the non-enzymatic function by degrading the whole protein, while as our SMIs are going to act by inhibiting only the enzymatic function of the target. So it is a limitation of our SMI, which this limitation is not applied to the PROTAC. PROTAC is going to degrade the whole protein, which is responsible for the disease. PROTAC have demonstrated particular sensitivity to the drug resistance target. While as SMIs are failed to address the confirmatory changes in the protein due to the mutation that leads to increase in a drug resistance. While as we already discussed that the protein of interest is being attached by our PROTAC, so the drug resistance can be overcome by using the PROTAC technologies. PROTAC have the potential to target the undruggable target. This is the most important part of the PROTAC, which the SMIs are not having. SMI does not having the potential to target the undruggable protein. SMI can only bound to the druggable targets. It cannot bind to the undruggable targets. PROTAC have shown the high potency. So as we already discussed that they are going to degrade the protein which is responsible for the disease. So that's why protein are going to show the high potency, while as when it comes to the SMI, it requires high drug concentration to maintain level of target occupancy that provides a sufficient clinical benefits. So more amount of drug is being required for the higher activity. So this is about the difference of the PROTAC technologies and SMIs. So as we already discussed in the milestone that PROTAC is now the under development. So these are the examples of some of the PROTAC which are under the clinical trials. Majority of these PROTACs are for the treatment of cancer, and they are in a phase 1 clinical trials. Some of the PROTACs are in the investigational new drug applications. Might be in the upcoming future, PROTAC may create a history in the drug discovery, and they can form a disasters in the therapeutic treatments. Next, we talk about the protein-based new technology. So we already discussed that the PROTAC is the bifunctional molecule, which out of which one is known as anchor and another is named as the warhead. So the two bifunctional molecule. So because of this two bifunctional molecule structure, there may be various technologies involved in the PROTAC. First, we are discussing the antibody PROTAC. Antibody PROTAC combines the specificity of monoclonal antibodies with the protein degradation capabilities of the protein technologies. They act by use the antibodies to selectively bind to the cell surface receptor or the extracellular proteins. Upon binding, they recruit E3 ubiquitin ligase to target the protein, leading to its degradation. Antibody PROTAC holds a promise in targeting cell surface receptors involved in cancer and immune disorders. They enable the degradation of the membrane proteins. They are challenging to address with the traditional small molecular system. Next, we are discussing the dual target PROTACs. So dual target PROTACs are designed to degrade the two different targets simultaneously. So firstly, we are discussing about a single target. So here, if there are two different targets, so this technology named as dual target PROTAC can be useful. They combine two different ligands, each binding to the distinct target molecules. When both targets are bound, the PROTAC initiate the ubiquitinase and the degradation. So that point should be noted here. Firstly, binding to the protein of interest is needed. So both our PROTAC molecule is going to bound to both the protein of interest. Dual target PROTACs can be used to modulate complex signaling pathways or the target multiple protein involved in the disease. So if there is more than one disease which is responsible for the if there is one or more protein which is responsible for disease, this dual target PROTAC can be having its role. Next is the aptamer PROTACs. So aptamer PROTACs use the RNAs or the DNAs aptamers as a targeting molecules, offering a different approach to the selectivity. Aptamers are selected to bind specifically to the target protein. When combined with a PROTAC structure, they can recruit E3 ligase for the protein degradation. Aptamer PROTAC provide an alternative strategy for targeting specific protein, including intracellular protein with a high affinity and selectivity. They have potential application in cancer therapy and other disease. Next, we are discussing about the OCTACs, which is autophagy targeting chimeras. OCTACs are the subclass of PROTAC, and they are designed to target a protein for the autophagic degradation. OCTACs recruits E3 ligase that tag the target protein with the ubiquitin-like molecules, directing into autophagosomes for degradation via autophagy. OCTAC offers a unique approach to clearing aggregated or the misfold proteins associated with the neurodegenerative disorders, another condition where autophagy plays a crucial role. Next, we are discussing about the TF PROTAC. So TF PROTAC is nothing but the transcription factor PROTACs, which aims to degrade the transcription factor which is involved in the disease process, especially in a cancer. So they target the transcription factors by recruiting E3 ligase, leading to the ubiquitination and the degradation, ultimately disturbing the downstream gene expression. TF-PROTAC have potential therapeutic application in the cancer and other diseases driven by aberrant transcription factor activity. So whenever the transcription are being involved, TF-PROTAC is going to play its role. Next, we are discussing about the LITACs, which is lysosomal targeting chimeras. So these LITACs are designed to target extracellular proteins for lysosomal degradation. They are the small molecular antibodies to bind to the cell surface protein, followed by the lysosomal trafficking and the degradation. So these LITACs offer novel approach to target and degrade extracellular protein, including those associated with the inflammatory diseases and cancer. Next technology we will discuss is the ClipTAC, which is chimeric ligase inducing protein targeting chimeras. So these ClipTACs are the variation of the PROTAC that induce the formation of ubiquitin ligase enzymes with the cells to degrade the target proteins. They promote the recruitment and activation of E3 ligase, leading to degradation of the target protein. ClipTACs have potential application in modeling specific protein functions within the cells and could be used for research and therapeutic purposes. The next and the last technology we are going to discuss is the folate case PROTAC. That is very emerging technologies in the PROTAC. Folate case PROTAC use folate receptor for selectively targeting. We know that the folate receptor is being involved in many processes. They exploit the high affinity of folates for its receptor to deliver PROTAC to the cancer cell or expressing these receptors. One inside, they initiate the target protein degradation. Folate case PROTAC show promise in targeting cancer cell with elevated folate receptor expression, offering a potential avenue for the cancer therapy. Kindly note that these are some of the technologies which are being involved in the PROTAC. I already told you that this is very emerging field, so there might be some other technologies available in this PROTAC field. So lastly, we are concluded as PROTAC technology signifies a transformative era in the drug discovery and therapy. It leverages the cell protein degradation machinery for selective removal of disease-associated protein. PROTAC addresses previously undruggable target, offering novel therapeutic interventions. So that point we already discussed that our SMIs are only going to target the druggable protein, while as our PROTAC can bind and degrade the undruggable proteins. It is a diverse PROTAC variation scattered to range of disease from oncological drivers to autophagy mediated protein clearance. Antibody, dual target, aptamers, AutoTAC, TF, LITAC, ClipTAC, and folate case PROTAC exemplify adaptability and the ingenuity. So the various technologies are emerged and they might be having a major role in the disease treatment. Ongoing research and clinical trial expand the scope and potential application of PROTAC technology. PROTAC promises to revolutionize therapy, minimizing side effects and improve patient outcomes. We already discussed that they are going to target the undruggable protein. They are going to degrade the protein which is responsible for disease. So by that way, the dose can be reduced off, the side effect can be reduced off, the cost involvement in the treatment can be also reduced off. They embody innovation and perseverance in the addressing complex health challenges. The horizon of possibility of PROTAC disease therapy is both promising and boundless, offering renewed hope for the healthcare future. So that part we already discussed. There is no end for this PROTAC technology. Whatever the disease causing protein we are going to find out, we can make a PROTAC for this, because this PROTAC is going to bound to the disease causing protein rather than inhibiting the whole of the receptors. So whole of the protein is being degraded by the protein technology, which cannot be possible with the available drugs. So this is about my PROTAC technology and it's unrevealing its potential. Thank you very much for the patient listening. If you are having any questions or if you want to collaborate in the PROTAC field, I mentioned my email ID here. I am happy to assist you. Thank you very much.